Hello and welcome to episode 582 of the People Behind the Science podcast. It is National Influenza Vaccination Week here in the United States, and we are hoping to help raise awareness and let everyone know that it is not too late to get your flu shot this year. So this week we are excited to be rebroadcasting our interview with Dr. Eva Harris, originally published on August 10th, 2014. Eva is a renowned infectious disease expert who has done important research on the influenza virus as well as the dengue, Zika, and chikungunya viruses. Her studies have spanned molecular virology, pathogenesis, immunology, epidemiology, diagnostics, clinical aspects, and disease control. And listeners, I enjoyed learning more about her life and her work in this interview. So sit back, relax, and get ready for another episode of People Behind the Science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to another episode of People Behind the Science. I am delighted to be here today with Dr. Eva Harris. Eva is a professor of infectious diseases and vaccinology, as well as director of the Center for Global Public Health at the University of California, Berkeley. She is also the founder and president of the Sustainable Sciences Institute. Eva received her PhD in molecular and cell biology from the University of California, Berkeley, and she completed a post-doctoral fellowship and served as an assistant adjunct professor at the University of California, San Francisco, before joining the faculty at UC Berkeley. Now, Eva has received a number of awards and honors during her career, including the McArthur Genius Fellowship, being named a Pew Scholar, receiving a National Recognition Award from the Nicaraguan Minister of Health, being named a global leader by the World Economic Forum, and also being awarded the Prytanean Faculty Award for Outstanding Women Faculty. Eva is here with us today now to tell us all about her journey through life and science. So Eva, today we are so excited to get to know you, both as a scientist, but also as a person. So can you tell us a little bit about what you like to do with your time when you're not doing science?
In those few moments.
Sleep, perhaps.
Yeah, well, I adore hanging out with my son, who is now 13, and we just have a blast together. And we just got bikes, and it's been so fun over the summer to spend even more time together. So that's great. My passion is dancing. I adore dancing, especially salsa, merengue, bachata, reggaeton. But it started from my hip-hop days and growing up in New York. So that's my hidden, or not so hidden passion. And every once in a while, when I actually get to read a book, that's kind of my nirvana from when I was young, before life hit. So.
Fantastic. The good old days of having time to read. I think I remember those fondly. So can you share with us today maybe a book that you have just loved over the years and that you would really recommend that our audience check out? It can be a science book or a non-science book.
Well, actually, the one that I have enjoyed recently, this is one that just came to my mind, but it's in Spanish, but it's called El Tiempo Escondido, The Hidden Time, and it's by Joaquin Barrero, and it's about the Spanish Civil War and then kind of spans about 4 generations as well. And it's a really fascinating kind of journey through history, through politics. And it's not about science, but I like historical fiction, but it's a really nice weaving of times and space in Spain, which I have done a lot of reading and spent a lot of time there. So I enjoyed that book, although that's not your typical book that you were probably expecting me to mention.
No, that's fantastic. I love hearing about these non-science books, maybe history books, mystery books, fantasy books, whatever people are out there reading. Because I think it's a great way to broaden your perspective. And I think history in particular is fascinating if it's woven nicely into a story. I think that's a great recommendation. So we'll put it out on our website for our listeners to check out on our recommended reading list. And I think that's amazing that you're able to do so many things outside of the world of science in that little bit of time that you have. You mentioned dancing, spending time with your family. That's wonderful. Can you tell us now a little bit more about your scientific side and how you describe what you do to someone who's not in science?
Well, really, I have 2 lives, and one of those lives has a lot to do with, I won't call it non-scientist, but definitely not purely academic science. And so I guess starting with the academic, you mentioned I'm a professor here at UC Berkeley, and my lab works on viruses, on dengue and influenza, mostly on dengue virus, which is a mosquito-transmitted virus, which is a huge global public health problem affecting about half the world's population, up to 100 million cases a year. So it's a big issue. And the approach that I've taken in my lab and beyond my lab through now 26 years of collaboration with the Nicaraguan Ministry of Health has been to approach the whole spectrum. So we look at the virus itself and its replication, host cell interactions, stress response, etc. And then we move beyond that into a mouse model, which we've developed, which allows us to look at both pathogenesis and immune responses and test potential antiviral candidate compounds and a number of different kind of bringing our in vitro work in vivo. But then we go beyond that into the collaborative work with Nicaragua. And there we have been running collaboratively a series of studies that, again, span from a hospital-based study to a clinic-based study called the cohort study, and then out to that into communities where we just finished a big randomized cluster control trial looking at community participation based on evidence as a way to control dengue through controlling its mosquito vector. And so that is kind of the breadth of the lab. And so we have active projects in all of those areas, including diagnostics as well. So that's kind of what I do day to day. Keeps me busy and everyone kind of along for the ride. My other life is more in the nonprofit sector, but I bring it into academia as much as possible. And that has to do with building scientific capacity in developing countries, especially focused on Latin America. And that I've also been doing for over 25 years. And so that led to founding a nonprofit institute called Sustainable Sciences Institute, which is headquartered in San Francisco, but also has a subsidiary in Managua, Nicaragua since 2004, and also in Cairo, Egypt for the past couple of years as well. And through that, we do a lot of work in training folks to address local, mostly infectious disease and other public health problems through lab and epidemiology and manuscript writing and proposal writing, and now a big area on informatics for health. So that's kind of the nutshell.
Right. Well, that is amazing. That sounds like so much to get done in a day, Eva. I'm impressed. So how do you do it? Is there maybe a quote or a saying that you love to keep in your mind, a mantra that keeps you focused and motivated to get through everything?
Well, it's not a very complicated quote. It's called siempre adelante, always forward. And that's what we say in Nicaragua when the going gets tough. So that's one, which I always think of just to kind of keep going forward. And the other, is hasta la victoria siempre, which is from Che. And that's, essentially always going forward towards victory. So it's just, again, kind of this moving forward despite any challenges. So that's kind of what I think of when I'm looking for inspiration.
Certainly. Well, those are two fantastic quotes. I love this focus on advancing, moving forward, pushing forward. And I think that's so important in science because it's easy to get bogged down in all the details, the day to day, and sort of just get stuck. So definitely pushing forward is a great motivational quote. Now, can you tell us a little bit about some of the people who may have motivated you along your journey or people who've inspired you and served as role models?
Well, one person who always comes to my mind is someone who I worked with in my undergraduate thesis. Actually, I was an undergrad at Harvard in biochemical sciences, and a professor from Switzerland actually came and gave a talk. His name was Jeff Schatz, and he gave a big plenary lecture, and I ended up getting to know him, and we just started talking, and he invited me to his lab, and I ended up doing my undergraduate thesis over the summer in Basel, Switzerland. And what really inspired me about him was that he was just a fantastic scientist, but he was also an amazing musician. And he also worked to kind of modify lab and other equipment to resource-limited settings, which is my first kind of view. I think he'd use like an aquarium pump to run a gel box and then I ended up doing a lot of that myself. But I remember just hearing about that, and I really liked the fact that he was a humanist as well as a scientist.
Well, that sounds like a fascinating role model, someone who kind of has a life outside of science, someone who does music and enjoys life outside of science, but also an amazing academic scientist, an amazing applied scientist in terms of transforming some of these technologies for use in low resource areas. I think that's fantastic. So can you tell us a little bit now about your journey and where it all began? How did you get excited about science in the first place? And what are some of these key moments in your life that you can remember that really brought you to where you are today?
Well, I think I've always been inspired by biology and the cell itself as a model for human society. That may sound funny, but I always mention this because I just think that the study of how the cell works, how molecules work together for the good of the greater whole, and then how cells work together for the good of the greater organism is so inspirational. And it's such a lesson for our own conservation in terms of both energy conservation and ecological conservation, et cetera. I think that it's really beautiful how biology has managed to figure out how to make it all work so beautifully and so efficiently. And I just think if, the human society could learn an inkling from what's going on in their own bodies, there'd just be no possible way that there could be the kind of conflict that there is today. So I've always kind of on that very broad level or, idealism level, love biology. But I like lab work. I like the process of discovery. I find it very exciting. My challenge was to bring that excitement to the real world as well. So, of course, I love the lab and the lab bench and bench science, but I always wanted to connect it in some way to what I call the real world. I mean, make an impact on life outside the ivory tower, as we say. And that was kind of challenging because when I was doing my undergrad and PhD, et cetera, that didn't really exist. Global health, as we know it today, didn't exist and certainly was not as trendy as it is today. And so everyone just thought I was crazy. and so I thought, well, maybe I'll merge I wanted to be a journalist and a scientist or something that would bring me out into the world. And honestly, during the time that I was getting my scientific formation or training was also in what we call the Reagan 80s, and there was politically very hot. I was at Harvard, and Harvard was invested in South Africa during the apartheid regime, and that was very upsetting to many of us. And so we were very involved in the divestment movement, and it was very exciting because while I was there, we thought that we had some influence, although it was probably the larger political and economic reality, but we're able to supposedly convince Harvard to divest from South Africa. That was very exciting. And also the Central America was very hot at the time with the Contra War in Nicaragua and the death squads in El Salvador. And so I was very involved politically, but then I also really loved science. And my vision was somehow to bring that together. And so that was essentially what I did, was to kind of bring my values to my science and trying to find a way to make bench science, pipetting, relevant to the rest of the world, as well as maintain a presence in an active program in basic science as well. So that's been my challenge. And then, kind of show folks how to do that in their lives as well.
Definitely. Well, I love that description that you gave of really being fascinated with the cell. It's sort of the blueprints for success are within us. They're all around us. Mm-hmm. So I think that is amazing. And really being able to merge that fascination with biology, with the cell, with society and how you can make an impact. I think that's incredible and great to hear about how all these little sort of historical incidences and things that were going on at the time really encouraged you to take that route of becoming active and being involved in this global health movement that's, I think, was just starting around then. So I think that's amazing. So can you tell us a little bit more about where you are now and some projects that you're working on that you are just so thrilled to be a part of?
So scientifically, there's a number of exciting projects. As I mentioned, we span the whole spectrum. So there's several different projects I could mention. Let's see. So one is on a kind of really a viral pathogenesis route that has to do with a dengue viral protein. Dengue virus is, as I mentioned, it's the RNA virus, and it's only around 10 kb of a single-stranded RNA genome and in enveloped virion. And so a lot of the pathology of the disease, which has to do with a syndrome that can lead to a vascular leakage and actually death within 24 to 48 hours of an otherwise healthy person, has been linked to what we call an immunopathogenesis, because there's these 4 flavors or 4 serotypes, dengue 1, 2, 3, and 4. And a known risk factor for severe disease is a second infection with a different serotype. And so the thought is that the immune response recognizes the current infection, but actually raises a stronger immune response to the previous infection. which is actually ineffective at controlling the current infection and instead exacerbates the disease. And so one project that we're doing has really taken me a long way because I was not trained formally as an immunologist, is essentially, moving, as I mentioned, from the virus and then to the mouse model and then into human populations. And we're really trying to understand the human immune response and how that can contribute to both protection and pathogenesis, and kind of pull that apart using this array of wonderful studies that we've been running for many years in Nicaragua. So we have a single study in a hospital, the National Pediatric Reference Hospital, that's been ongoing now for 16 years. And so we have the same protocol, a lot of the same doctors, et cetera, so that when something changes, both clinically or epidemiologically, it's not because the protocol changed, it's really because something real is happening. And it allows us to kind of dissect both the clinical aspects, but then also bring it back to the virus and to the immune response. And then we can also link it to a different type of study that we run, which is a cohort study. And in that case, that's actually now in its 11th year. So it's the single longest continuous study looking at dengue in children, where we recruited around 3,500 children in 2004. And a lot of those same children are still in the study. And we essentially look at healthy children and then follow them for when they get sick with dengue, but then also with many other diseases as well. And that's really exciting because it allows us to look at the samples that we take every year prior to their dengue infections. And so we can tell which children got infected and got sick, which children got infected in the same year and did not get sick, so controlled their infection, and then go back in time to their prior sample to see what is it about the immune response that could be correlated with this protection or conversely with the pathogenic response. And so that's exciting to actually bring the immunology and the virology into human populations. And it's very challenging because naturally, like, humans are not inbred like mice.
Right.
There's like a gazillion variables. So it gets kind of messier and messier the further from the lab bench that you venture out. But it also becomes, all the more relevant. And so that, I think, is the constant tension between like reductionist and the relevance and where you fit on that spectrum. So that's like one kind of major area of the lab. Another is just this very specific project on a particular viral protein called non-structural protein 1, NS1, which is the only viral protein that's secreted. So there's a short period of viremia where the virus is circulating in the bloodstream for about 5 days. And right when the viral load goes down, which is when your fever, it's an acute febrile illness, goes down, that's when you have this potential period of shock and death. And it turns out that this viral protein, NS1, also follows that same curve and has only been looked at to date as a marker for viremia. But also, it's been thought that antibodies, NS1, may play a role, or NS1 does a lot of interfering with the complement cascade and immune evasion. But we've very recently discovered that that protein actually can act directly to cause vascular leak and endothelial dysfunction. And we've shown this both in our mouse model and in a in vitro model with primary human pulmonary endothelial cells. And so it's really, really exciting because it's a completely new function that is really important for the overall pathogenesis of the disease. And so we're working really hard to understand the mechanism and, what are the parts of the protein that are important and what is the part of the cell that's involved, what's the signaling and all this. So it's been very exciting to kind of take that deep into the cellular and virological mechanisms.
Certainly. Well, Eva, those sound like fascinating projects. And I think so far we've talked about all the good things about the world of a scientist, just the amazing work you get to do and how you're able to really combine it with your other passions. And we mentioned one of the struggles that you have just in terms of not having enough hours in the day. But I think a lot of scientists run into just different struggles and major failures that they hit along their journey as scientists. And I think oftentimes they may have weeks or maybe even longer where it just seems like everything is going wrong. Now, Eva, can you take us back to a point in your career where you were having a major struggle or perhaps a major failure and tell us what was going on and what are some of the steps that you took to get through that really tough time?
Yeah, I think one of the hardest times, I guess, in my professional or academic career was really when I decided to kind of come back to academia. So I had as I mentioned, I did my undergrad at Harvard in biochemical sciences. Then I did take time off. I took a year off, which is when I traveled a lot around the world and spent a number of months in Nicaragua, which is where I initially connected and decided that I would work there for, in part the rest of my life. But I also came back then and did my PhD in yeast genetics, as it turned out, very basic science and chemistry and molecular biology. But after that, I was already starting to work on this vision of bringing scientific tools to developing countries. And so I then took a hiatus because I just felt, even though I'd had postdocs set up, I felt that I couldn't just turn my back on all this really exciting work that we'd started. And it was very inspirational to many folks around the world because these were very, very new techniques like the PCR and new molecular biology techniques, applying them in country like where there was barely running water, barely electricity, and actually addressing local health problems. It was just so incredibly exciting. But no one was doing it at that time, making that connection in the sciences, in the molecular biological sciences at any rate. Then that just swept me in this kind of snowball. I had no idea where I was going, but I just went with it. But I realized that I needed some kind of a resource base after a while, 5 years of this, and kind of existing with a completely virtual program of people who are supporting me and working and coming and doing workshops all over the world for free, but eventually I needed a resource base, and I didn't see myself in industry or working for government. And so the only thing was to essentially create a research program. But to do that, I needed to come back to academia. And I had always been connected. It was all while I was doing a postdoc at UCSF. So I still had a foot in either world. But figuring out how to take on the challenge of starting a brand new research program, starting a new vision, a paradigm that was going to really bridge from the lab to the field, which didn't exist, coming kind of back to this country. And I ended up having a baby at the same time. And it was very overwhelming. And
could life get crazier, right?
and as you can probably tell, I never, stick to the beaten track, but this was way off it, right? And so most people will, have a track record in a particular organism or a particular area and will have a number of papers. And, and the next step is you get hired into an assistant professorship on that area. But I had kind of completely changed from yeast genetics, into I was studying maybe 5 different infectious diseases in 10 different countries. And that isn't your general standard profile for an assistant professor. Mm-hmm. But I ended up getting this McArthur Award and then was recruited to UC Berkeley. And I decided, okay, I'll come back, but I need to decide on what to focus on. And the reason that I ended up on dengue was because everywhere that I went in Latin America, there was many countries that I was working collaboratively with. I said, what are your biggest health problems? And they said, well, tuberculosis. Many said leishmaniasis, which I was actually also working on, and then dengue. And I mean, I didn't even know how to spell the thing. And then to my shock and horror, it turns out to be a virus. And, I was completely eukaryotic. I mean, my yeast genetics, parasites, my whole vision was around, genetic screens, or microbial pathogenesis. I mean, this virus stuff, it was completely incongruous. I had no idea how to start thinking about viral pathogenesis. But I felt that I had to put my money where my mouth is. And if that was the biggest public health problem, there were so few people addressing it seriously at that time, I just felt I had to take up that challenge. So it was a brand new, not just organism for me, but it was like my virology, immunology, clinical, statistics, epidemiology. It was all completely new fields, and so I kind of had to just jump in and make it happen. So that was indeed challenging. But it was very exciting because I had this whole program going and had attracted, by virtue of people who were interested in it, wonderful, wonderful scientists who wanted to kind of be part of starting that journey together. So I had some fantastic postdocs and folks also from Latin America who were part of my initial crew. And so that working in solidarity against this huge challenge was kind of how we pulled through. But that was scary at times.
Definitely. Well, that is an extraordinary story and amazing to hear about how you were able to put all the pieces together. Like you said, starting in a completely new, essentially, field, learning everything, putting everything together, getting this research program together. I think that is amazing. And I think really inspirational that you were willing to go out there, talk to people, say, what is the biggest problem? Oh, something I know nothing about. All right, well, we'll fix that. Let me figure it out and make it happen. I think that is incredible. So definitely a challenge turned into a success. So we don't just want to talk about these tough times today. Can you tell us, Eva, about another one of your successes in the world of science, something that's meant a lot to you?
Well, again, there's one that's a science and one that was part of the scientific capacity side. But I think one of the challenging and funnest experiences was a project that we did that was led by a postdoc in the lab, but that really, truly combined multidisciplinary research, bringing together the fields of virology, immunology, phylogenetics, clinical research, biostatistics, epidemiology. And it was essentially solving the mystery of this increase in dengue disease severity that we noticed over this one period of time, which was from 2005 to 2006 and 7. And again, because we have these ongoing studies for many years, when something funky happens, we know that it's not just that we switch doctors, that there's actually something real that's going on. What's really interesting is that there was this big increase in disease severity in both our hospital study and in our cohort study, which are very, very different study designs. And so we ended up collaborating to sequence the virus and to do a lot of multivariate analysis to try and figure out what was driving it. And the only thing that was different was the year. And the only thing that happened in the year was that it turned out that the virus actually shifted clades. So as I mentioned, the virus has 4 serotypes, but it's like Russian dolls. Within the serotypes, there are genotypes, and within the genotypes, there are clades made up of different strains. And so the clade shifted right over this time. And so initially we thought, that must be it. And the initial statistics that we did showed that there was a direct relation with the viral clade and disease severity. But then the postdoc and I, her name was Molly O'Ainley, she was just fantastic. And we just couldn't rest easy because we knew that we had done full-length sequencing and we had only been able to capture about 75% of the virus isolates. And so we said, well, what about that other 25%? And we've got to go after that. And so we developed a genotyping method. And when we actually were able to cover the rest of the viruses, our whole hypothesis fell apart because there was no longer a statistical association. And we were like, oh, no. And so we had to kind of go back and say, well, what else could be involved? And it turns out, of course, as I mentioned, the immune response is very important in dengue because your immune response to a previous infection makes you at higher risk for the current infection in terms of disease severity. And so we went, and that actually did explain part of it, but it didn't explain everything. And so the long and short of it was by combining like all of these different methods and data sets and techniques and people, we were able to come up with the hypothesis that it was actually the serotype-specific immunity in the children combined with the changing viral genetics which had actually been responsible for this increase in disease severity. And then that was based on large numbers of severe disease in children in our hospital study. And then we were able to come up with a hypothesis and directly test it with samples from our cohort study by going back in time and choosing pre-infection sera and then being able to actually test that hypothesis against the outcome of the disease. And in fact, it turned out that children who were essentially immune to dengue 3 ended up being higher risk for a dengue 2 infection of the second clade that emerged, whereas children that were immune to dengue 1 were more at risk for severe disease with the first clade. So it was fascinating to understand the dynamics of these two processes and to also understand that it's only when you really address that massive complexity that includes time and space and human beings and a lot of people collaborating together that you can really unravel that complexity. So that was really exciting. It was a long process, but it was really satisfying to be able to kind of come finally to some kind of conclusion that addressed what I felt was like the true complexity of the question.
That is a fascinating project and an amazing success to put all the pieces together, figure out the complex interactions between, like you're describing, sort of the immunity, the virus, and everything that's going on there. Now, can you compare it at all to like the influenza experience here in the United States, where, it's more of a seasonal thing? I know transmission is different between these two viruses, but is it something where these shifts in dengue are common, or are they pretty uncommon? Or, you expect it to be the same every year and it's abnormal when it's not?
Well, so it's interesting. So dengue spreads through mosquitoes, but it also is carried geographically. And the geographical introductions happen because of travel of people for the most part. So people will become infected. Many are inapparent infections or asymptomatic infections. Will get on a plane, get off somewhere else, and then be bitten by a mosquito, Aedes aegypti or Aedes albopictus, locally in that new place, and that will start a new epidemic or a new introduction. And so it's really the movement of mosquitoes and then the movement of the virus itself, which makes essentially the constant reintroductions of dengue at the level of a serotype or a genotype. But at the level of the clade, there's this constant movement of clades being replaced, of dying out, of coming more. And that's actually another whole question, is like, what drives that evolution? And interestingly, we actually went back to that same story, and the question was, is it actually viral fitness, like something that the virus actually replicates better in the clade that replaces the previous one? Or is it completely stochastic? Is it random? Is it just, okay, this one happens to be there then? And so what we did is we went back and we actually took these very similar viruses, the clade 1s and we call the clade 2b, and competed them together in primary human cells and saw that there was a replicative advantage for the replacing clade, which was interesting. And then we went back and we was asked, well, would that also be true in mosquitoes? So I went and we collected Aedes aegypti mosquitoes in Nicaragua around the time that this epidemic had been going on. And then we brought them back here to the States. I carried them several times through. And then we collaborated with wonderful colleagues who are entomologists. And then someone in my lab learned how to handle the mosquitoes and do all these complicated experiments. And we actually were able to show that when we took low passage viral isolates into low passage real Nicaraguan mosquitoes, that we actually were also able to show that there was a small but consistent advantage of that replacing clade. So we actually think that there's a replicative advantage, which is kind of a fitness advantage, which was driving this replacement. So it's kind of different in the sense that the thing about dengue is that these are complex. And so it's rare that you really can drive down deep into understanding why each of these, say, clade replacements or disease severity increases occurs. There's many, many examples, but very little evidence about what has actually driven the change. Whereas in influenza, for instance, we know that there's just a shift, it's a resortment. Although there's, of course, a lot of fascinating science in terms of how those arise and, what the specifications are of the new virus, et cetera. But it's slightly different than in dengue, but there's similar concepts, but because you have a mosquito involved as well, it makes everything even more
adds another layer there. Yeah. Well, that is really interesting. And it sounds like kind of in talking about these different research projects that you've worked on, you have been able to do so much amazing travel. Mm-hmm. And I think this is one of the things that a lot of scientists point to as one of their favorite things about the career, to be able to experience this international research culture. Now, can you think back on your career as a scientist and think about all the places that you have traveled for science and share with us today your favorite travel experience or your favorite place that you've traveled to, and tell us a little bit about why it's so important to you?
So there's two kinds of science travel that I do. One is for conferences, both domestically and internationally. And so that's always fun to some extent. But I would say that, in my particular history, the most memorable, if you wish, experience has been my 26 years of working in Nicaragua, because that, was a very powerful experience the first time, because it's the second poorest country in the hemisphere. And it was very different from my conditions growing up. I grew up in New York and Paris and, not in a particularly wealthy environment, but certainly I had a house and roof over my head and food on the table. And,
you're comfortable.
A comfortable middle-class existence. And so the kind of extreme poverty that was kind of the day-to-day reality, and immersing myself in that was really an eye-opener and really made me even the more committed. I mean, I already thought I'd been committed, but this was like really committed me to say, okay, we got to change this world, and I'm going to try and use science to do that and make it a better place for everybody. So even though at that point I was also traveling in many countries in Latin America particularly, and you could name a couple that I really love, like Colombia and Ecuador. My husband's family is from El Salvador. But I think that, the kind of staying with this, even though the political situation changed many times over the past 25 years, but I stayed with my colleagues and staying and working in Nicaragua, and that's been really amazing. I go there maybe 6 to 8 times a year still. And so that continuity has been it's like a second home as well. But I also think that the travel, much as it sounds fun, is really very difficult, especially as a mother. And I just have to say how much it's incredibly painful for my son. I'm sure my husband too, but I particularly feel towards my son and his growing up. And I mean, I bring him everywhere I can. He comes to Cuba, to New York, to Chicago, everywhere he possibly can come with me. But obviously, as he gets into school, it's harder to take him out. I mean, he's in middle school and high school, and it just doesn't get any easier. It's just so painful for him always when I'm not there, and for me as well. So I just think that, the travel is fabulous on some level. But it's really, really difficult for especially a mother, I think. So I just have to put that out there.
Absolutely. Well, I'm so glad you shared that with us today, because I think that is something that doesn't often get said when scientists are talking about, weeks on end where they're bouncing from place to place all around the world, is that they're leaving their family behind for that time in a lot of cases. I mean, like you said, you try and bring them when you can to certain conferences and things, but ultimately when you're out there doing field work, you can't necessarily be with them, whether they're at the same location or not. So I think that is so important. And it's great to hear, though, that you were able to spend so much time down in Nicaragua and really get to know the people and really get immersed in the culture and really get an appreciation for the life of the people there. I think that's incredibly powerful to see and really be immersed in. And I think one of the amazing things about being in this world of science is the people that you get to work with. So I think over the years, when you look back over these different research groups and maybe projects you've worked on and different labs you've worked in, you just remember fondly the people as just amazing and creative and oftentimes very funny people that you get to spend time with. And looking back, oftentimes there's these quirky traditions that spring up in different research groups, or maybe just really funny memories you can look back on fondly. So can you share one of these with us today?
Well, actually, I was thinking more in relation to not a particular question per se, but just, I like to work hard. I also like to play hard. As I mentioned, I like to dance hard. So whether it's in Nicaragua or here, this is a joke, but kind of the litmus test for joining my lab is if you like to dance all night. and,
is this the initiation?
Yeah, exactly. And actually, it's kind of a joke, although of course in Nicaragua it's not, because everybody, goes out a lot. But here it's fun. We have a group now that really likes to go out dancing salsa, and the most unlikely people hit the dance floor. So that's really fun. But of course, not as often as one would like. But there you go. And I think one thing I just wanted to mention, in terms of the people in one's lab, et cetera, I think that in my vision, it hasn't just been me going to Nicaragua. Initially it was kind of me holding together this whole program, this spectrum of activities. But as time has gone on, my vision has really been scientifically to go from the lab to the field and the field to the lab, in other words, to observe what's going on in human populations and then to try and dissect those or understand what we see in the lab, both here and in Nicaragua. And conversely, to come up with hypotheses in the lab that can then be, tested observationally through our studies in the field. But also it's in terms of people themselves. So there's a number of Nicaraguans in my lab now. And over the years, we've trained, maybe 15 or 20 Nicaraguans here in my lab. But I also have a graduate student from Ecuador who's just joining, and then from Brazil and other places. So I think that that's important in that direction. But also many other graduate students, postdocs, undergrads, pretty much along every level, technicians as well, have come down to Nicaragua. So I think that really broadens one's appreciation of science and also of the specific scientific questions when you can place them into an epidemiological and clinical and human context. So I think that that's an important aspect of the humanity of the lab itself.
Well, I think it's great that your students get that opportunity then to go to where the research questions are, not just kind of you working with these abstract models and theories and the equations that they're putting together in the lab, but actually being out there and seeing the impact of their research is. I think that is incredible and so important in science. And I love that you teach your students that and the humanity of it all as you describe it. And I have to ask, you mentioned you have a lot of international students coming in the lab. If students come in without dance skills, do you provide them lessons?
No, but they come out and they learn.
Gotcha. Trial by fire there.
No, but you don't have to be a perfect dancer to join the lab. So there you
Gotcha. Well, that is amazing. And I think that is a great tradition to have go out, have fun with the lab members, go out dancing, and certainly to bring them with you out in the field, as well as bring people from those countries that you're visiting back to the lab to learn from you as well. So that is amazing. Now, we've spoken a little bit about these research questions that you're working on answering, both in your academic life as well as in the more nonprofit sector as well, things you're working on. But we haven't yet talked about some of these unanswered questions that are burning in your mind. So can you tell us today, Eva, if things like funding and staff and technology and feasibility, the normal constraints on your work, were not an issue, what is one question that you are just dying to know the answer to?
So one thing that I've become really fascinated with, it kind of has to do with that tension between the reductionist and the relevant, if you wish, that I was talking about before. But I remember when you're in the lab, you're looking at a particular protein and you're looking at a particular situation, and you try and minimize the variability so that you can actually conclude something and repeat the experiment with the smallest error possible. But you're also constraining so much of the environment to do that. And that's just in the lab. Now, if you take it out of the lab into an animal model, then it gets trickier. And if you go from that into human populations, it gets even trickier. But, even so, it's very challenging to look at, say, dengue or influenza. I didn't even mention this huge influenza program that we have in Nicaragua in collaboration with one of my former students, Aubrey Gordon, who's really pushed this whole influenza area too. So that's really exciting because we can look at dengue and then in a kind of dual cohort with influenza. But it's actually not just about two diseases. I mean, folks in countries where disease is endemic are actually infected with many different pathogens. And those pathogens influence their immune system, and it influences their response to other pathogens. And so I think it's really fascinating to me how multiple infections will influence the outcome. And it's so narrow-minded of us to pretend that, this person is infected with dengue and what's their out, which is what we do. I mean, that's how
it's
else
so convenient. You only get infected with one thing. Yeah.
You have to constrain the question in some way. And it's hard enough that you're out in human populations, but it's also false. I mean, I know already that the helminth infections are changing, with the Th2 response. I know that their IL-10 is up. And I know that even the outcomes that I'm measuring are not just because of dengue. I know that there's other things going on. And so there's now with these we call it the multidimensional omics. I mean, there's so many levels that one can gather information, at the level not only of the actual pathogens or, through metagenomics, et cetera, but also through the microbiome, which is, our own organisms within organisms, which we already know now in this fascinating new field, how much that can direct the outcome, not just of disease, but of course, obesity and many other aspects that we didn't know were being driven by our microbes. But that's another whole level. And then there's also just kind of the individual signatures like EQ3L mapping. And so we've actually started a number of these sorts of projects, or your own host genetic components, which also will influence. So one of my dreams, and we've actually started doing this, it's collaborating among folks who have a number of different systems biology approaches and try to kind of bring together people around particular really well-defined cohorts, like, for instance, ours. This is my dream. We have 11 years of data on these children. We have the ability to go forward in a collaborative manner with both Nicaragua and, of course, our study participants, and be able to try and really broaden our vision to understand the complexity, not just of one pathogen and another, but even the larger picture of, what is behind the outcome analysis that we do. We have one nice story where we actually were able to look specifically at the interaction between dengue and influenza, and then we made an observation in the field. We brought it back. We made a mouse model. In fact, this is true. We do get more severe disease, when we have influenza followed by dengue. And so now we're looking at the gene expression analysis of that. But even beyond that, to kind of even broader. So that's my big vision that I would love to be able to address in some way over the years to come.
Certainly. Well, those are some amazing questions. I think when you start talking about all the different combinations of things that could happen, it's sort of the possibilities are endless. And I think all these different layers of complexity make it even more fascinating, but even more difficult to study, certainly. So that is an incredible question, and great to hear that you're starting to make progress towards putting some of these complex, more sort of in vivo, in real life scenarios together. Now, I know over the years of scientists' work with many amazing people, as we mentioned, and I think oftentimes you're inundated with sage advice from so many different sources. Now, can you think back, Eva, on all the advice that you've received over your career and over your life? And can you pinpoint just one piece of advice that you would consider one of the best pieces of advice you have ever received, and share it with us today?
Yeah, so again, this is very simple. Just to try to remember to breathe every once in a while. Literally, that's simple.
Let's pause right now. Yeah.
When you're overwhelmed, you just look out the window and actually take several breaths, and I realize I'm not breathing. And then that's very calming. It actually could, just a couple deep breaths. And it's funny because I think about it, and every time I think about it, I realize, I'm actually not breathing right now. I have oxygen coming in every once in a while, but just to take a deep breath and then that's actually some of the best advice I've ever been given.
Well, I think that is amazing advice.
Yeah, not very complex, but it's probably one of the things that comes to my mind as the thing that's the most useful. That and to try and sleep every once in a while as well, but that I'm not as good as taking that
Breathing's
advice.
a little easier to do than schedule sleep. So,
Exactly.
well, I think that's amazing advice. And sometimes it is just these simple pieces of advice that really stick with you. Just remember to take a moment to breathe, to relax, and then you can keep pushing forward and moving forward. So that is amazing advice for our listeners. Is there any last piece of advice, Eva, or last piece of inspiration that you would like to share with our listeners today then from you personally?
Well, I guess kind of to follow one's passion and not feel constrained by the quote unquote beaten path. I guess I'm lucky in the sense that the beaten path actually frightens me. I'd actually rather be out there with the machete, trying to make a path through the jungle. But I think that sometimes I see in students, with good reason, I mean, it's a scary world out there, especially the funding situation these days. But I think that if you really follow your passion, people are drawn to that. And I think that that's kind of one of the most important pieces of advice. And also I think really important in these days is also to bring values to your work and to your science and not feel that your work or your academic life has to be divorced from your value system. And I think that bringing that in is actually really enriching and inspirational to people. And so I would say that that's actually a really important aspect as well.
Absolutely. Well, those are two more amazing pieces of advice, Eva. And I think today you have shared with us so many insights and amazing stories. Can you now tell our listeners a way for them to reach out and contact you if they want to learn more about you or your research? Sure.
Best is my email, which is eharris, so EHARRIS at Berkeley, BERKELEY.edu. And I would just say to be persistent because as we discussed, I'm often traveling and it may take a moment to get back, but I'm always remembering who's written me. And if you write again, I will be sure to write back.
Gotcha. Well, thank you again so much. It was wonderful to speak with you today.
Well, thank you.
Thank you again. And listeners, thank you for joining us as well. We'll see you next time on another episode of People Behind the Science.